Hi guys, I just wanted to go over the sensation and perception slides really quickly with you because I updated them and I'm not sure if you noticed it, but I put a new one up and just updated some of the pictures. But I want to point out what's important for the test. So as we go down the slides, one, when you look at this picture closely or quickly, what do you see? You see a rose, and then if you look at it in more detail, spend a little more time, start looking at the little pieces, you'll see a couple that's hugging and kissing here. You can see their legs hanging down. You can see a stocking thrown over here, a stocking down here, shoes kicked off. So you can see a little bit of different things going on when you look closer and you start to break it down to the details. So in this chapter, we're going to talk about bottom-up processing and top-down processing. So what you did when you looked at this and just saw the rose in a picture frame or a window type thing, then what you did was top-down processing. When you looked at all the little elements and looked for the things that I'm pointing out, you were doing bottom-up processing. Normally, when we engage in sensation, those are the things that fall on our receptors from the external environment, we use bottom-up processing. When we engage in perception, when we're adding meaning to what we're receiving on our senses, and we interpret it and we organize it, we're using top-down processing. Okay, so first we're going to talk about sensation. I want you to realize that sensation and perception are two separate things. So sensation is that physical stimulus coming in, for example here ringing a bell, and it's still physical at this point. So the physical stimulus of hearing is a sound wave. So the sound wave is funneled into our pinna, and it goes through all the mechanical, physical things, and it gets to what's called the point of transduction. Every physical stimulus that we have coming in eventually gets turned into a neural response, or you could kind of say a neural stimulus. So the physical stimulus turns into a neural response or pulse, and it goes to the higher parts of our brain, to the cortex, and we perceive a bell. So the sensation is the physical sound wave. The experience of the ringing of the bell is our perception. Okay, absolute thresholds. This is the smallest amount of a stimulus you can detect half of the time. So sometimes you might hear it, sometimes you might not. So here's examples of absolute thresholds. We can actually see a candle if there's nothing in the way, no trees, no people, no cars, no buildings, 30 miles away, 50% of the time. Hearing, we can hear a mosquito 10 feet away. So you can go through each one of these and see these are the absolute thresholds. These are the smallest amount it takes for us to detect them reliably 50% of the time. Subliminal stimuli is things that come in unconsciously, out of our conscious perception. So at one time there was a movie theater, and the very first experiment with this, where this man said that he flashed pictures up too fast for the brain to process. So it was subliminal. It wasn't processed or perceived by the brain. Yet it was hitting our receptors at some point. So he said when he flashed food up, it caused his sales to go up at the theater. But it turns out that nobody could replicate these experiments, and he finally admitted that he really didn't do the experiment properly. Okay, so let's go to the next one. Let me make sure that's next. Okay. So this is another case study. So there's thresholds. And threshold for frequencies of motorboats, these animals, they're manatees, cannot hear them. And so these manatees, normally, they're very gentle creatures. I lived in Florida, so I saw them many times. In fact, they came up in my parents' backyard, and we used to feed them lettuce. It was pretty cool. So they come down where the warmer waters are. They're really gentle giants is what they're called. And they are actually an endangered species because of their inability to detect the sound of motorboats. So when those air pressure waves go through the water, they do not hear them. And this is an example you can see where he was sliced up by a propeller that came by. So we have very strict laws in Florida, no wake zones wherever the manatees are. So you have to go very slow. Okay, difference threshold. Now, I want you to understand the difference between an absolute threshold and a difference threshold. So the absolute threshold, remember, was the lowest amount of stimulus we could detect 50% of the time. A difference threshold is actually the difference between two stimuli. What is the lowest amount of difference that we can detect? So I want you to keep the weightlifter in mind. If she's lifting, let's say she's got 75 pounds. Let's say she has 3 of those 25s on each side. So she's got 75 on each side. She has 150 pounds altogether on that, and whatever the bar is, right? So like 10 pounds for the bar. So 160 pounds. And if we added 5 pounds to each side, she probably would not detect it. Because relative to what she's already picking up, it doesn't make that much of a difference. See the word difference in there? But if we added another 25 pounds to each side, she would definitely feel that difference. So it is the amount, the smallest amount that a stimuli can differ, and we would pick it up. Okay, so signal detection theory says, look, there's really no such thing as absolute thresholds. We are really, our determination of how we detect something is really our motivation behind it, the state of mind that we're in, the elements of the experience or the environment at the time. How motivated are we to detect the stimulus that we're trying to detect? So this is an example of if you were a nurse and you had to detect tumors, you would probably be much more sensitive. You would probably have a lower threshold for detecting them because it's so important. So you can see the ones that are in green, those are good. You definitely want to detect it if it's there. You definitely don't want to detect it if it's not there, because the alternative is you could tell somebody they have cancer, and if you're wrong, you just put somebody through, which would be the false alarm, undue emotional trauma, right? But if you're wrong, if you miss it, oh my gosh, somebody goes away thinking they're fine and they actually have a tumor developing. So there's a lot that goes into whether we detect something or not. Let me put this a little more at home. If you're home alone, you ever been home alone? Nobody's in the house at all? Nobody. And it seems like those times, especially if you have to spend the night in the house alone, say your parents go out of town or something, and you hear every little noise outside, you swear it's a footstep, right? You can hear crackling of leaves. We become heightened. We become more sensitive when we're alone at home because it's a little scarier. I know it's not totally scary. You're in your house, you're safe, but there are times when we're home all alone and we swear we hear something. You're a little more sensitive to it. So this is just another example of it. And there it is, the definition. So we detect things according to our experience, our expectations, our motivations, and how alert we are. So like I said, when you're home alone, you're a little bit more alert. Or if you just watched a scary movie and you're home alone, even more so, right? Because you have an expectation, you've just had an experience, you're very much alert because this movie has kind of set you in that tone. Okay, so attention. What do we pay attention to? Do we pay attention to everything? No, we don't. We can't. We can't possibly pay attention to everything. But that is what is the difference between sensation and perception. Everything in the room around you right now, if you just roll your eyes around the room and scan it, and then look down, tell me what's in the room. List to yourself what's in the room without looking at it again. So as you scanned around the room, everything in the room hit your visual receptors. It did, but you did not perceive it all. If you keep looking down at your computer right now and don't look back around the room, you're not going to remember, even if it's your house that you've lived in, you're not going to remember every detail of what you just scanned. And if you do it in a strange place, it's even more so. So we are selective in our attention. So we don't notice all the details, and that's a good thing. We don't want to focus on every detail. If I saw every detail everywhere I looked, I would not have time to think or focus on what's important. So what happens is we end up trying to focus on what's important. And in this slide where it says missing the gorilla, and this is a picture of the Disney castle in Orlando. So my example is that I've been to Disney World over 100 times in my lifetime. I literally have lost count. I've had season passes. I go several times a year. I've gone since I was a child. I've gone with my school every year with my parents, and I've taken my children several times. So I have several visits to Disney World. I have looked at this clock so many times. I don't know how many times. I adore the castle, and I've taken several pictures of it. I never noticed that this clock has on it not just the time, but it tells you the rotation of the moon and the sun and where it is at any given time during the day, as well as all the zodiac signs and where the planets are. All in this one clock. I never noticed it till I studied it for this lesson. The missing the gorilla part is an experiment that people did where they gave people a task in a circle. And as they were doing the task, a gorilla, a man dressed in a gorilla suit, came out and waved behind them and then ran out of the room. The people were so engaged in the task because it was competitive that they never saw the gorilla in the room. So we are selective with our attention, and it is for a good thing, okay? Multitasking. I'm sure a lot of you think you're good at multitasking, but when we divide our attention, we are not as good as if we focus our attention. The point is, you might be good at multitasking. You might be able to listen to music with words in it, study, watch, and then watch a movie all at the same time. I know kids that do that, or young people that do that. And they think they're good at it, and they might be able to pick up the gist of the movie and still get stuff done and pay attention and text and do all these different things at once. But in reality, research supports that we are better when we focus our attention and we don't divide it. So you take that for what it's worth. Okay, so let's talk about the visual system. I'm not going to test you on amplitude, and I'm not going to ask you, okay, which one is for color, which one is for brightness, but wavelength is for color. The longer the wavelength, as for example, red. Red is a very long wavelength. So that's why we see tomatoes as red. Tomatoes reflect a very long wavelength, so we perceive them as red. In reality, tomatoes are gray. Tomatoes are a shade of black and white. Everything is really a shade of gray or black and white in our world. Nothing has color. Color is what our mind adds to it. It's in the mind. And I know that's hard to believe, but it is. Why do we all perceive the same color on a tomato? Because we all have the same basic setup in our eyes, which is perceives the length of a wave, of a light wave. Okay, so the visual stimulus for the visual system is light. So these are light waves. This is the electromagnetic spectrum, and I want you to notice that right here, this little sliver is all we perceive. All of this is not in our perception. So yes, this color range is all right here. We don't see ultraviolet. We don't see infrared. Now, if we use special glasses, we can. We don't see the microwaves in the microwave going around. What do dogs see? They don't see black and white. They do see color. They see blue and yellow. All right, the anatomy of the eye you are going to have to know. You're going to have to know the pathway of light. Light travels through the cornea. That's this gelatin type structure that goes around the outside is the cornea. The light goes through the cornea. The next thing it goes through is right here. This is the color part of the eye, the iris. That's a muscle. Light does not go through the muscle. It doesn't go through the iris. In the middle of the iris, the colored part of your eye, is an opening. That opening gets smaller and bigger. As it needs more light, it opens, it dilates, it gets bigger. As it gets too much light, if you're out in bright sunlight, it constricts It contracts, it gets smaller, so it lets less light in. But that's what light does. It goes through the cornea, it goes through the pupil, and then it goes to the lens, which reflects the image that we're seeing upside down on the retina. Okay, on the retina or in the retina are the receptors. So if I'm asking you a test question, I'm going to ask you, what is the pathway of light? You're going to tell me the cornea, the pupil, the lens, and then the retina. Okay, where is the point of transduction? It's in the retina. The receptors in the retina are the point of transduction. You need to know where the points of transduction are. That is where the physical stimulus goes to this bottom layer of the retina and it stimulates those cells. And those cells stimulate, and those are rods and cones. And then they stimulate the bipolar cells. And then they stimulate the ganglion cells. And those cells, the axons, travel right here out of the optic nerve. This is the only place on your retina that doesn't have receptors because it's where all the axons leave the eye. That's the optic nerve. This is your blind spot. Okay, now, if we laid out the retina, let's say this is the strip of retina all laid out here. Where you see the red are the colored receptors. Those are cones. They're all right around the very center, right here, the back of the eye. So all the information coming straight in here goes to the middle of the eye, and those are your cones. That's where most of your cones are. Your cones are your color receptors. Rods, you can see how you have much more out to the sides, away from the middle. So all out here, you've got the congregation of rods. Rods are what you see black and white with, okay? And here's the optic nerve. So this is the blind spot, which is very close to the fovea, all right? You do need to know the difference between rods and cones. One is color, one is black and white. One is more congregated towards the middle, one is more towards the outer parts of your retina. Okay, you need to know the pathway. This is the pathway to the cortex. Okay, this is what happens after the physical stimulus is transduced into a neural pulse. Okay, so it leaves the optic nerve. So the optic nerve is first. Then you get to the optic chiasm. See, right here. It's like an X. So I think of cross, chiasm, it's a crossing. So this is the optic chiasm. And you'll note that the information from the inner parts of the eyes, the inside of your eyes, closest to your nose, cross over to the other side. But the information from the outer parts of each of your eyes, they stay on the same side of the brain. These pathways are called the optic tracts. Okay, so they follow these optic tracts, and then they go through the lateral geniculate nucleus, which is in the thalamus. Okay, so you've got your thalamus here, and then the final destination is the occipital lobe back here. Okay, so it goes to the optic nerve, back of the eye, to the optic chiasm, right here. It follows the optic tracts to the thalamus, and then goes to the occipital lobe. You need to know this pathway in this order. One of the tricks that I keep to memorize the pathways, remember that they all start with the nerve that's leaving whatever sensory system it is. And they all have the thalamus right before they get to the cortex or lobe, which is your cortex, okay? So the thalamus comes right before. There's only one system that the thalamus is not part of, and that's your sense of smell. Okay, the trichromatic theory of color vision is this. Oops, sorry. We have 3 color receptors in that first set of cones. Okay, so when the visual image comes in and hits our receptor, the first thing it does is it hits 3 different cones. We have blue, green, and red. Now you'll look at this and say, well, how the heck do we get yellow? Because you've been taught your whole life that the primary colors are blue, red, and yellow. And depending on how you mix them, you get all the other colors of the rainbow. Don't think of this as mixing paint. That's the difference. These are receptors for wavelengths. It's not like mixing paint on a tray. That's why more red and green together actually add up to a yellow perception. So that's not the end of the story, though. We actually have two systems that make up our perception of color. So after these receptors send their information to the brain, higher up in the brain, we have what's called the opponent process system or theory, and that is where we have receptors or neurons that fire to, like a light switch, okay? So when the light switch is up, it's one color, and when you put the light switch down, it's another color. So you have the opponent process, the opposing color. So it's like a color wheel, and it's whatever color is opposite. So we really have receptors that respond to yellow and green. So I'm sorry, yellow and blue. So on one side is yellow, and if you flip the light switch the other way, it's blue. On the other one, it's green and red. So if you flip the light switch towards green, the other side is red. So if you stare at this, and I'm going to give you a minute to stare at this, or let's count to 30, okay? Just stare at this dot right here in the center of this flag. Let's do it together. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22, 23, 24, 25, 26, keep your eye on the dot, 27, 28, 29, 30. Now shift your eye over to this one and you should see the American flag. If you do it for 1 minute, you'll see it much clearer. But the point is that you just saw the evidence of the opponent process. Okay, so you were looking at the green and yellow, and it was fatiguing your receptors. They were on the switch for green and yellow. The opponent of yellow is blue, and the opponent of green is red. So when you look at the white space, it flips to the other side, the opponent colors, because you've been staring at these colors so long you fatigued it. So when you looked over here, you saw the other colors. So that's evidence that we actually have an opponent process going on. You can do the same thing here, and this is what happens with people who are colorblind. If they don't have the neurons that respond and fire and give the information of green, then they won't see the number in here that you see. Because you have the vision for both, if you're not colorblind, you should see the number in here, which I think from what I'm looking at is 74. Okay, and over here it's the opposite. If you don't have the perception of red, then you would only see a green circle here. You wouldn't see the 8. Okay, you can do it, you can practice this with your textbook actually, because here you can see too much. But if you like cover these and you just stare at the red for a while, you'll fatigue the red to where you don't have the capability of seeing red. And when you look here, you won't see the 29, it disappears. And vice versa, you can do it the other way. Okay, so when you look at this, and actually when you look at this, okay, when we're deciphering visual information, we can look at certain details, or we usually look with top-down processing. So right now you're either going to see an old man or you're going to see a young man. Everybody's different. Like we said before, signal detection theory does hold true. Some of you might be in one kind of mood, some of you might be in another. You might have just been with your dad, you might have just been with your boyfriend, you might have been with someone younger, older, whatever. So it determines what you see first. Some people see the old man, some people see the sun. So I'm going to point it out. If you only see the old man, now I want you to shift to your feature detection, which is bottom-up processing, and I want you to strain to look for these features. So let's say you see the old man. Here's how you see the young man. This is the old man's nose. If you're trying to see the young man, that's his chin. This is the young man's chin, and he's looking off to the side. This is his ear, this is his chin, and this is his neck. And he's wearing a bandana, and he's got a cowboy hat on. Now, if you saw the young man and you're struggling to see the old man, realize that this is the old man's nose, this is the old man's eye, and this is the old man's mouth. Okay, and he's got an older hat, not really a cowboy hat, it's a little different, more of a floppy fishing hat sorta. Okay, so the old man or the sun, whichever you saw, you can struggle to look at features and see the other one. That's your bottom-up processing at work, and this is how we know we have feature detectors. So this was actually Nobel Prize research. And Hubel and Wiesel did this research where they put electrodes in cats' brains and they showed them slanted lines, they showed them horizontal lines, they showed them moving horizontal lines, and different parts of the brain would light up for each of the different stimuli. So we know that we have feature detectors specific for things in our environment in our brain. Okay, so let's go on to hearing. Once again, I'm not going to test you on amplitude and frequency. I think of amplitude, you can remember that it goes with loudness, because think of an amplifier. When you turn it up, it's louder. So that's how I tell the difference or remember the difference between the two. Okay, but take note of the ranges of hearing. Human hearing is right in here. So you can see that ultrasound we can't hear, but bats can hear and dolphins somewhat can hear. And here there's sounds that are out of our range as well, like an earthquake. We don't hear an earthquake coming, but we have seismology information and machines that can detect them. Now look where the dog is, and this is the interesting thing to me. He's right in the range of human hearing. I don't know if you've ever been sold one of those whistles, dog whistles that humans can't hear but dogs can. They don't work. It's a scam. It's a scam. Okay. You need to know the physical pathway of the stimulus, the physical stimulus for hearing, which is a sound wave. It's a pressure wave. It goes through the outer ear, which is called the pinna, and it goes through the ear canal to the eardrum, or the tympanic membrane. Most of us know it as the eardrum. Tympanic membranes, they're tight, yes, yet they can vibrate, just like a drum head. When you hit the drum with the drumsticks, it vibrates. It causes movement to these three inner ear bones. They're called the ossicles. The ossicles are actually the hammer or the malleus, and the incus and the stirrup or the stapes. Okay, so when these move, they move like a lever system. This one moves, causing this one to pull back, which causes this one to pull back. So right now it's still all physical, right? It's still movement, physical movement, and this is your middle ear. Now, when this is pulled back, it causes the fluid. This is a fluid-filled little snail-looking thing here, and it's called the cochlea. It causes this fluid to move like a wave going around. So the next slide unrolls that snail. Okay, so if we rolled it straight out, this is what it looks like. It's a canal going around this basilar membrane. And it comes to the circular window. This is an oval window for the stapes, and this is a circular window. So and it's kind of like, think of it like a rubber bandish or a rubbery type of material. So when the wave comes crashing this way, it expands and it takes in that pressure and then sends it back this direction. Well, while this fluid is going around, it's touching this basilar membrane, and right here are cilia. These are hair cells on the basilar membrane. It causes them to bend. So that's where you get the point of transduction. So the point of transduction is located in the inner ear, and the point of transduction is actually when these hair cells, these small tiny hair cells called cilia, are bent or move. That's the point of transduction, the cilia. Or if I ask you which part of the ear the point of transduction is in, it's in the inner ear, because that's where the cochlea and the cilia are. Okay? Here's the pathway for the neurostimulus. Once it's not a physical stimulus anymore, once it's past that point of transduction, here's the pathway. It's the auditory nerve. So that's remember where all the axons leave, make up the nerve, they bundle together and they leave. They go through the medulla, then the midbrain, then, like I said before, the thalamus is always the last step before the cortex, which for the hearing is here on the sides of our heads. The temporal lobe is on each side of your head right around your ear, which makes sense because the information that's coming in is auditory. So that makes sense. Okay, oops, gosh, this moves so easily. Okay, so I want you to take note here of the decibels that are safe. So normal conversation is 60 decibels. When you put those iPods in your ear, the little earbuds, you're hearing things way over what's really safe for you. 100 decibels is a lot. and but we don't listen to it for enough hours at a time that it could, well, it could damage your hearing. Here, this is like a rock concert, 130 decibels. This could actually damage your ears. Okay, this I left in because we have things where we can just bypass if the physical features are not working right. We can go right to the neural pulse and stimulate it. So we do have these devices now. I don't know if you've seen on Facebook, sometimes they have the little videos of a baby who hears his mother's voice for the first time because he gets these cochlear implants. So it's nice. We are progressing, or as a people, not we, not me, but researchers are progressing with things that may actually get rid of deafness someday. That would be great, wouldn't it? Okay, so let's go on to touch and taste and smell. Okay, so somatosensation. If you remember from chapter 2, you have a somatosensory cortex, and that's a part of the brain that is right next to, so if you look at the lobes, if you go back to chapter 2 and you look at your lobes, you've got your frontal lobe and your parietal lobe. Along the parietal lobe, along the central fissure that divides those two lobes, there's a strip, and it's our sensation, it's sensation cortex, somatosensory cortex. So let me show you. So this guy right here is the homunculus man, and it's very interesting. I wish I could get a model of these, but this picture's online. Each part of his body represents how much of that somatosensory cortex, so that strip along the brain, the top of the brain there, how much of it is dedicated to each part of our body. So processing that information. So you can see a large part of our cortex is dedicated to processing information from our hands and fingers, which tells you you have the most sensation right here. You have the most awareness. You are going to perceive this information better than, say, your shin. So tell me, what part of the body is going to cause more pain if you hit it with a baseball bat? Your shin or your hand? Definitely your hand, because more of your brain is dedicated to sensing and perceiving it. So that's just interesting. Okay, so we have several different receptors for pain and touch. You do not have to memorize the names of these receptors, but notice this is why we can feel such a huge range of feelings, physical feelings, because we have such an array of receptors that detect all different things. Like this one detects stretching. Okay, this is Ruffini's endings. It detects when someone's pulling on your skin or stretching something, pulling your hair, anything that causes stretching. This is what it picks up. This picks up pressure. This picks up deep pressure. So when you're getting massaged, these free nerve endings are picking up pain. So the hair follicles, all these different give us this combination of feeling. We have such a wide range of feeling. The gate control theory, I don't want you to think of this as an actual flap opening and closing along your spinal cord. That's not actually how it works. I'm not going to hold you to being tested on all the factors of the gate control theory. And if you were in my face-to-face class, I would map it all out and draw it for you. But the point is that we actually have interneurons that are keeping our pain messages from going to our head. And when pain comes in that we need to feel, it stops inhibiting those and allows them to send a pain message to the brain. So really it's not so much a gate as much as it's an interneuron that's constantly inhibiting our pain receptors until we actually feel pain, and it stops inhibiting because they're inhibited by the new pain message coming in to allow the pain receptor to send its message to the brain. So that's actually how it works. Endorphins respond to pain and pleasure, and here's how they work with pain, and this is very interesting. So in chapter 2, we learned how neurons fire, and they don't fire until they reach threshold, which comes from the top part of the neuron, up here. These dendrites send little electrical pulses down here, and they're added up. So they're pluses and minuses, whether they're excitatory or inhibitory. And when we get to a point of threshold, once it all adds up to say +3, so I have plus I have 6 positives coming in, and I have 3 negatives coming in. So boom, I have a total of +3, which is my threshold for this neuron, and it's going to fire. So it releases neurotransmitters down here at the end. This is the end of a pain message coming in. It's stimulating this receptor. It's releasing neurotransmitter, which is going to tell this receptor it's feeling pain. The interesting thing is that endorphins bypass this system, and instead of them releasing neurotransmitter on dendrites and giving these pulses of inhibitory pulsing to add up to inhibit the cell from firing, they just come right down to the end and say, stop releasing your neurotransmitter. That's different from a normal neurochemical function. So it shows how they can respond quickly to pain and relieve us. If you want to, you can watch this video. It's just grooming chimps. And the thing that's interesting with this research is that, oh, excuse me, they found that when they studied them in their natural habitat, they groomed way longer than they needed to be clean. So they don't just groom for cleanliness reasons or keeping themselves hygiene reasons. Let's put it that way. They actually do it because they like it. That's what it seems like, because they do it longer than they have to. So there has to be another reason they're doing it, because it feels good. When endorphins are released, we feel pleasure. That's why we like massages. We like back massages, foot massages, someone brushing your hair. So as you watch these monkeys, it's kind of cute, because one point, the one that's being groomed, he's like, no, get me over here, a little bit over here. And he points to his other shoulder, and the monkey moves over there. And one of the research projects that was done on this was pretty cool. They showed, they were watching these 2 chimps that were grooming each other for a long time. This one chimp gave this other monkey like way longer grooming than he needed. So when it came to be snack time, this monkey that had groomed him went over in his little clique in a different part, and this one was in his part, and they all had their snacks, and it was snack time. It wasn't their dinner, what they needed for sustenance. It was like, it's like when you get a treat. It was a treat time, right? So as he's eating his little snacks, he had one left. So the monkey who received the grooming took his last treat and ran over to the monkey who gave him all that nice grooming time and offered his last treat to him. So that kind of tells you something about it, that it's a little more than just hygiene. Our kinesthetic sense is just our sense of where we are in relationship to the world and where our body parts are in relationship to our inner core. So this system right here is made up of these 2 things, vestibular sacs and our semicircular canals, which kind of look like pretzels. And these tell us where we are in rotation. So if you go on those rides that turn upside down, or like dizzy rides that make you sick afterwards, it's because of this. You get a little dizzy, things are spinning. In here we have crystals in our vestibular sacs, and they have, when they get out of alignment, when they get out, we feel very dizzy, like we can't walk. We don't, our relationship of our body to the earth is completely out of whack. And this, you may have experienced this if you've ever had vertigo. And the problem is getting those crystals back in place, and you will not feel well until you do. Okay, so smell, this is the one system, like I said, that doesn't have the thalamus as a pathway. So I want you to see that the pathway is the olfactory nerve. It's always the nerve, and it's always going to the cortex. So the one thing in between is the olfactory bulb. And then we have the frontal lobe. So it goes right up here, the information. So see how quickly this goes to the frontal lobe of your brain? That's why cocaine is so addicting, snorting it at least. All right, so taste buds, taste, our sense of taste. Here's the pathway for taste. The first thing it hits is the tongue. And then, and remember, these 2, smell and taste, are the chemical senses. So it's a chemical, not an air wave, not a light ray. It's a chemical that comes in, and it filters in, and actually the chemical itself stimulates the receptors, stimulates that first nerve cell. It goes to the medulla, then it's projected to the thalamus, then the parietal lobe. Okay, so now let's talk about perception. Going through these different things, we're really focusing more on top-down and bottom-up processing. Remember, I told you top-down processing is where just the overall, what we see at first, and then when we break it down to its elements, break the whole down into its pieces, that's our bottom-up processing. That's more breaking down all the little sensations. So let's go over some of these. All right, so here we're talking about top-down processing. When we first look at this, I see 12, 13, 14. When I look at it this way, it's ABC. But if I took away the A and the C and I only showed you 12, 13, 14, you wouldn't think, oh man, that's a B. No, you'd see it as a 13 because that's your top-down processing. Now, when I tell you, can you see this as a 13 and it's a B? Yes, but you have to do your bottom-up processing to see that. Now, same thing here. If you look at this picture, the first thing you see with top-down processing. It's quick. It's a way for us to look at things. And we see an old man. We see a face of an old man with wrinkles and a hat on. But when we break it down, we see two men standing right here on each side, and they're kind of wearing a robe-like thing. And then we see a tree going up the middle, and then we see rows of, I think, branches of the tree, and we see birds in the tree. We see horses. We see a horse head here. We see a horse head here. We see a horse head here. We see a cannon here. We see soldiers here and here. We see a leaf sticking up here. So all these different things are in this picture, but at first glance, we just see an old man looking at us. Right? Same thing when we look at this one. You can either see a very young woman here or you see an old lady. Okay, so if you see the old lady, this is her nose, this is her mouth, this is her chin. But if you see the young lady and you have to break it down to flip it to see the two different pictures, this old lady's nose is really the young lady's chin. And this is like a strap she's wearing around her neck. And this is her ear. So you can see one or the other. Same thing with this one. You first see two old people looking at each other and maybe a vase in the middle of the two of them. But if you look at it, you've got trees, you've got a bridge up here, and this becomes a waterfall. And then you see two, I think, Mexican men on each side. One's playing a guitar. This one's holding a jar on his head. Here's an empty bottle of booze here. This becomes a curtain. These become rugs, and this is a person in the doorway instead of an ear, and this becomes a curtain instead of hair. So you can bottom up process and see the details, or you can top down process and see the two old people looking at each other. This is another type of illusion, and if you stare at this enough, it should move. And this one also, if you try and stare at one point, it moves. So it's kind of cool. All right, let's get into Gestalt principles. This is figure ground. So whatever comes to the foreground is the figure, and whatever sinks back and is just a backdrop is the ground. So if you make the vase figure, then the black is just a background. But if you make the vase background, move it to the back and bring the black forward and make it figure, you see the Queen Elizabeth and Prince Philip. Okay, another principle, Gestalt principle, is similarity and proximity. So the law of proximity is that they're close to each other, so they must be a group, basically. But if they're very similar, you could also say they're a group. So let's say I asked you how many groups are here. You might say, well, there's two. There's two groups of circles. They're similar. Or you could say maybe there's just one. There's just circles because everything's similar. They're all circles. But proximity tells me I could say this one is close together and this one's a little further apart. It's a group of fours. So again, the law of proximity is these two, this is closer, so there's two groups. However, I could see this as 8 groups, right? Because we've split them up. So those are those laws. Continuity. Do you see one continuous line here? At first glance you will, but if you struggle, you can see that there's 2 continuous lines. So there's a W. I'm going to help you out here. There's a W in the middle, right there. That's one line, and then this is this line. So there's actually 2 lines. We perceive it as one because we see things that are the simplest. We tend to see the simplest explanation, and we tend to see things as continuous. We also tend to see closure. So you can do this with words, you can do it with pictures. Sometimes when you look at words, you're going to see things that you might read a whole paragraph and not realize the word the was left out a couple times. You just filled it in. Same thing when you're looking at a picture. This is not blobs of black. We see a panda, even though it's not a complete panda. We see a star here, even though there's no star. There's Pac-Men and little triangles. That's all there is, but we perceive a star. Okay, common region. So we can, if I asked you how many groups are here, if you went with the law of similarity, you could say 2. There's circles and there's stars. But this one trumps it when you see region, okay? So we see 3 regions. You would say there's 3 groups here because they're in a closed region together, even though they're 2 different types of symbols, okay? Depth perception requires binocular cues and monocular cues. That's the only way we can see depth perception. So someone who's lost an eye cannot perceive depth perception. Monocular cues don't require both eyes. So one eye will perceive these. So what I'm showing you here is the linear perspective. As parallel lines converge or go forward, it looks like in the distance that they come together, like when you look down the road. Okay, relative size, in our minds, we don't perceive this man as a giant, not at all. We perceive them as both probably about the same size, but what we perceive is this man must be closer because he's bigger. There's a bigger form falling on my retinas. The physical stimulus is bigger, but I don't perceive him as a giant and, oh gosh, there's a 9-foot giant. No, my mind organizes the information and says, this guy's closer to you. Same thing with interposition. So what's falling on my retinas is horses overlapping. And when objects overlap, my mind constructs them as being closer. So whatever overlaps the other one, I see it as closer and my mind perceives it and explains it that way to me. Light and shadow, this is really cool. We tend to perceive light coming from above. So when our mind organizes the information, what's falling on my receptor here, these look like they're bubbling out. They look like they're towards me. These look like they're concave in. If this was a domino or a Lego and I flipped it, I just flipped it over, turned it over 180 degrees, these would appear to be sticking out and these would appear to be sinking in. It would be this exact same picture, but they would switch. These would no longer look like they were popping out if I flipped it over. Texture gradient is where things go away into the distance, they become smoother. So, and the things that are closer to me appear close. The things that are more detailed appear closer. So this is the reason right here that you shouldn't take a selfie very close up, because if you have any flaws on your skin, any texture, it's going to show. Further away, it all smooths out and looks good. All right, aerial perspective. Do you see how hazy everything gets back here? So when something gets hazy, it's perceived or it's constructed in my mind as being further away. Motion parallax is when, as we are close to, so let's say we're looking far away at something far from us. So she's looking at the sun. She's riding in a bus, I guess, or a car, and the sun's way away. It appears to move in the same direction with us because it's far away. However, things that are close to us appear to go in the opposite direction. So our mind perceives things that are close, things that are going the opposite direction to be closer to us. If it sees an object that's further away, if there's an object further away from us, our mind interprets it as moving in the same direction, so it's further away. Okay, so binocular cues is convergence. So we see things with our eyes from 2 slightly different positions, and that makes sense because your eyes, if you close one, if you look at something in the distance, so pick anything in the room, look ahead of you, pick an object, close one eye, and then open the other and close the other. The object appears to move over a little bit. That's the retinal disparity. It's the difference between the left and the right retina. And all that information is going into the brain to help us perceive things correctly. Okay, so some of the constancies that we have, even though we have something different falling on our retinas, we still perceive them to be the same, unchanged. The perceptual constancies are these four things. We perceive size not to change. We perceive shape, brightness, and color to be constant, to remain the same, even though different amounts or different colors or sizes are falling on our retinas. We don't perceive them as changing. We perceive them as being constant. So I love this example here, and I always have to backtrack it to really see it. Okay, so these are located here. So these are, when you go up here, they're blues. And we perceive them as blue when we look at this picture. Then you come down, then if you pull it out of the yellow, here's the actual color, and when you pull it out here, it's gray. Now, take this same box and put the lens of blue over it, okay? I still perceive this as blue. And then when I pull it out of here, the actual color is gray. In both pictures, I'm sorry. I did this wrong. This is yellow. These are the yellows. We perceive them as yellow. Break it down. And this is actually the yellow squares. So this yellow up here is really this color, the same color here. But when we see it over here, we perceive blue. We see it over here, we perceive yellow. So even though they're not the right colors, actually, in actuality, they're gray, we perceive them as yellow and blue. Is that not very interesting? Okay, so let's look at the other one. All right, oh, and let me just say one more thing. So out of these, right? So like the guy who was so tall, we don't perceive him as changing in size or being bigger. We just perceive him as being closer. Same thing with shape. When I look in the mirror, so if you look in the mirror right now, you see how wide you are. If I turn sideways, what's falling on my receptors is a much skinnier me. But I don't get all excited in my mind saying, oh, I've suddenly gotten skinny. No, I perceive my shape in my head as still being the same, even though what's falling on my retinas is different. Okay, these illusions, so what they illustrate to us is that what falls on our brain is so different from what we actually interpret, right? So this line here is the same length as this line, but doesn't it almost feel impossible? It feels like no way. That has to be a longer line. But if you want to measure it, you'll see they're exactly the same. Same thing as here. This line looks so much longer, yet we still perceive it as being bigger, but yet it's not. What's falling on my receptor is the same exact size line, and it is so hard to see that. Okay, this room is famous, right? This is an illusion that someone built this room to make it look like this guy is totally giant and this guy is midget, but in actuality, if you look down at this line right here is actually coming forward like this. So this guy is much closer to us than this one is, but it doesn't look that way because of the way they created the room to give us that illusion. Extrasensory perception. If you go down here, you'll see all the definitions for each one of these different types of ESP. And research, unfortunately, does not support this. There's been maybe a couple cases where maybe it indicates, but for the most part, it shows us that this can't be, it's not true. So that's all I'm going to say about that. I'm not going to test you on these definitions. Okay, this is an example of, and this illusion is that when you grow up in a culture that is totally different, like for instance, we grow up in a culture where we have square buildings, straight lines, everything is real precise, manufactured. When people grow up in cultures like this where things are rounded, they do not fall for these illusions. They actually can tell that the lines look the same to them, whereas for us they don't, because we've grown up in a culture, in a world where we've become accustomed to interpreting things a certain way. So that's all I wanted to say. I wanted to go through these slides with you because I'm worried when you take the test if you're going to remember the things that you should. And what is important. Okay, so if you have any questions on anything, give me a call or text me or send me an email, and I look forward to seeing you all on test night.